right, science friends. Today we are talking about how genes are expressed. So remember, every cell type contains the same genome. It contains all of the DNA, but every cell type does not express every single gene. And even for the genes that it does express, it doesn't produce those genes all the time. Okay, so let me give you an example. The genes that code for your eyes do not produce hair. Like, we don't need hair growing out of the middle of our eyeballs. And so those eye cells do not make the gene for hair. And so that's just kind of a overly simple explanation. You have enzymes that break down things like lactose, like in milk. But if you're not drinking milk and you're not eating dairy, then you don't need those enzymes. And so it would be wasteful for an organism or the body to make those enzymes. And so we have to have ways to regulate these pathways. And so that's what we're talking about here in this first slide. This is an example of a metabolic pathway. And the example that this is shown is creating the amino acid tryptophan. So this example that we're going to walk through here and use as a model, you don't have to remember this example specifically, but I'm just going to use this to model the process. Is for E. coli. So E. coli is a bacteria. It has to have tryptophan to survive. And so generally, these E. coli bacteria are going to try to acquire tryptophan through their host. But if the host has not a lot of tryptophan, then they will start to synthesize their own. And so this process is regulated by two mechanisms. The first one is regulation of the enzyme activity. And so this is an example of feedback. So we talked about feedback in Unit 1 and then again in cell communication. So in feedback, basically we have a product such as tryptophan that's going to stop its own synthesis. So remember, feedback can be positive or feedback can be negative. And so in positive feedback, the response amplifies the stimulus. In negative feedback, the response stops or reduces the stimulus. And so that's what this is here. This tryptophan production pathway, as the tryptophan builds up, that's the response because we're making more and more of it, it can stop its own synthesis. And so this is in negative feedback. Generally, things that are negative feedback are, the goal is to bring them back to homeostasis. And so more often, things that happen in living organisms are an example of negative feedback because we're trying to restore homeostasis. Okay? Now, another way that we can regulate a metabolic pathway, this should say B here, is regulation of enzyme production. So we have to regulate that gene expression. And so this control happens at the transcription level. So remember, transcription is taking DNA to RNA. And so if we want to turn off a gene, essentially we don't want to make the RNA for that gene. We will stop the C's transcription, and as a result, that protein won't be produced. Okay? So this product, tryptophan, is made by this cell metabolic pathway or cell signaling pathway. So you have this precursor molecule that is broken down or catalyzed by enzyme 1 to make this, and then you've got enzyme 2 to make this, and you've got enzyme 3 to make the tryptophan. And so each of these enzymes, remember all enzymes are proteins, and so they are coded for by a gene. And so if we want to stop this process, another way besides feedback that we can do that is stop the production of those enzymes. If you turn these genes off, then enzyme 1 won't be produced. And so this pathway would stop, okay? And so regulating gene expression is also an example of how we can regulate metabolic pathways. And again, Both of these are an example of negative gene regulation because we're going to try to stop the process here. Okay? So today we are going to talk about operons. And operons, it is really important to know, so write this down, they are only in prokaryotes. Okay? Only in prokaryotes. So what is an operon? Well, an operon is a segment of DNA, okay, that has a series of several genes on it. And they're controlled by one promoter sequence. So you see here we have the promoter, and we have several different genes, tryptophan E, D, C, B, A, and that's coding for all of these enzymes like you see here in this process. And so it makes sense that if in this metabolic pathway we need to make all three of those enzymes, and it takes 5 genes to do so, that all of those are going to need to be either all made or all not made, okay? And so in the DNA, these genes all are controlled by the same promoter. And so when polymerase accesses this operon, it'll make all of those genes because they're all needed for that metabolic pathway, okay? Now, again, this is only something that we see in prokaryotes, that those genes for any one specific pathway are all right there together, okay? Because remember, prokaryotes only have one chromosome. For us, it's different because we have lots of different chromosomes. And so the chromosome 1, the gene for one part of a metabolic pathway may be coded for on chromosome 1 versus the gene of another metabolic pathway may be coded for on a different chromosome. So it's a little bit different for eukaryotes than prokaryotes. But again, this operon includes the promoter and all of the genes that are within that promoter sequence, okay? So let's talk now about the operator. So what the operator is, it is also within this promoter sequence. It controls the access to that DNA pathway, okay? So the operator is like the on and off switch. It's either going to say, yes, RNA polymerase, you can access these genes and you can make all of those, or no, RNA polymerase, I'm going to turn you off and we are not going to transcribe these genes right now, okay? And so that operator is, again, the switch, all right? Again, this is on the DNA, and this is RNA polymerase, the enzyme, which you should remember is involved in transcription, taking that DNA into the mRNA, okay? When the operator allows RNA polymerase to access it, it will go on and it will make tryptophan E, it will make D, and it'll keep going all the way down the strand. And all of these polypeptide subunits that are needed in this pathway that we see here will be made, okay? Now, we can, though, however, control this operator and turn it off. If there's plenty of tryptophan in the cell, then the bacterial E. coli cell does not want to waste its energy to make the tryptophan. And so in that case, there's a molecule that comes up here and binds to the operator and turns it off. And so now polymerase can't go because it runs into this big roadblock. And so it can't access those genes. And so those genes would not be made, okay? Those proteins would not be synthesized. All right? So there are 2 types of operons that we're going to talk about today. Repressible operons and inducible operons. So the difference in these is that repressible operons are usually on, but they can be turned off, okay? Inducible operons are usually off, but they can be stimulated or turned on. And so those examples, as you see there, you don't have to memorize those, but the tryptophan and the lac operon. So the lac operon is where these operons were first discovered. It was discovered by two scientists named Jacob and Monod. So sometimes you'll hear it referred to as the Jacob and Monod model. Okay, so let's take a look into both of these. So repressible operons have a molecule, or a protein rather, called a repressor. And that repressor binds to the operator, and so then polymerase can't access those genes. And so transcription will be blocked. The genes will not be made, okay? Again, repressible means that they're usually on, but it's going to inhibit them. So the repressor binds to the operator and blocks the transcription, turning that transcription of those genes off, okay? Now, the repressor is also regulated by a secondary molecule called a co-repressor. And those two together have to work to turn it off. The repressor can't do it by itself. It has to have the co-repressor bound to it to make that happen. Now, if you think back to cell signaling, what frequently happens when two molecules bind together is you have a shape change. And that's what happens with the co-repressor. When the co-repressor binds to the repressor, you have a shape change. And we'll talk about that in just a second. Okay? Let's take a look here, though, at this in the middle, these regulatory genes. So what is a regulatory genes? Well, repressors are proteins, and so they have to be coded for in transcription and translation as well. And so the genes that code for the repressor are known as regulatory genes. Now, generally, we are going to have repressors in low levels in the cytoplasm at all times, okay? But that's not a problem because, again, like I said, you have to have the co-repressor to actually activate the repressor to be able to stop the transcription. And I'm going to explain that a little bit more in just a second. Another thing that's important to know is that repressors are specific to a particular operator. Okay? A repressor is only going to turn off one operon. It's only going to work on one pathway. Okay? So it's very specific. Kind of like enzymes and substrates, they only work, it has that lock and key fit, so to speak. Repressors are kind of the same way. Okay? So let's look at an example here and kind of see what I'm talking about. So here we have our operon. Remember, our operon is made up of all of these genes that we want to transcribe together because they're involved in the same metabolic pathway. We have our promoter sequence. Within that promoter sequence is the TATA box. That's what RNA polymerase is going to bind to. And then also the operator. Okay? Right now, RNA polymerase has full access to that operon. And so in this image, all of those genes would be transcribed. And you see them being transcribed here in the mRNA, and then translated here to those polypeptide subunits. Okay? So at this point, the repressor is inactivated. Again, remember, this is a repressible operon, meaning it's generally on, but we can repress it. We can stop it. And so in order to stop it, what you have to have is you have to have a co-repressor that binds to the repressor molecule. So this is a repressor molecule. Here at the top, it's inactive, okay? That repressor molecule does not fit in that operator. Those two puzzle pieces do not fit together. These point out, right? The operator points nice and up and parallel. And so because that doesn't fit, this repressor is inactive, and so the operon stays on, okay? Now, when that co-repressor is present, the co-repressor here is tryptophan, okay? going back to this pathway that we're talking about here. The tryptophan, when it builds up, it's going to cause the gene to be able to be turned off, okay? As you have excess tryptophan start to build up in the cell, it's going to realize, hey, like I'm wasting this energy making more tryptophan. There's plenty of tryptophan here. And so it's going to act as its own co-repressor for that process. And so that co-repressor is going to bind to the repressor, and you have a shape change, which activates it, okay? You see that shape change there? Now, this activated repressor can bind to the operator, okay? It's going to bind to the operator, and polymerase is blocked. Polymerase can't access the operons now, okay? It can't access those genes. And so the operon is turned off, no transcription happens, and so we're not going to make these proteins for tryptophan synthesis, okay? If tryptophan is present in the cell, we don't need to make those. If tryptophan is absent in the cell, then we need to make that, all right? And so that is an example of a repressible operon, okay? This is pretty in-depth. I'm trying to explain it as best as I can here, but it's probably something that you will need to revisit again, okay? If you thought transcription and translation was hard, operons are just, a little step further. So go back and revisit this after we talk about it today, all right? So that's the repressible operon. Let's take a look at the other kind. The next type is an inducible operon. So the repressible, our example was the tryptophan. The inducible operon, our example is the lac operon. And so the lac operon is lactose, is referring to lactose. So lactose is available to E. coli through the host's diet. And so when lactose is present, we need this enzyme called beta-galactosidase that's going to break down lactose into a simpler sugar molecule, into a disaccharide, okay? Now, if there's no lactose present, we don't need to make this enzyme. But if lactose is present, then we need to make this enzyme. And so this is an inducible operon. It's generally going to be off, okay, when lactose is not present. But when the cell has lactose present and we need to break it down, then we want to induce it. We want to turn this process on, okay? So an inducer is opposite of a repressor. It is the small molecule that binds to the repressor, and it's not opposite of repressor. It binds to the repressor, which binds to the operator. Okay, I said that a little bit incorrect. So we still have the repressor, but instead of a co-repressor, we have an inducer. Okay, so in repressible operons, you have the repressor, and the co-repressor binds and stops it. In inducible operons, you have a repressor, and an inducer binds and allows it, okay? So here again is our repressor molecule. That looks the same as in the other one. The inducer is going to bind to that repressor molecule and inactivate it. And so now this is going to not fit in that active site, in that operator. When that repressor is active, it fits in the operator, okay? Once the inducer binds, it inactivates the repressor, and it causes it to fall out of the operator. When it falls out of the operator, then polymerase can access those genes. When the repressor is, when the inducer is not in the operator, then that's going to block polymerase's action, okay? So a little bit different here with repressible and inducible operons. Now, The last type of thing that I want to talk about is an activator, okay? So an activator is a protein that binds specifically to the DNA. So here's our activator molecule, and it's going to bind to the DNA in the promoter site. Here's the key here. When the activator is present, it makes RNA polymerase more likely to bind and transcribe. When the activator down here is not present, RNA polymerase is less likely to bind and transcribe. And so activators help stimulate transcription of a gene, okay? All right, so what I want you to try today is you have an assignment. It looks like this. I called it an Operon worksheet, but it doesn't have a title on it. So this is what it looks like. There's about six scenarios that I want you to walk through. And for each one, you have to say if it's a repressible operon or an inducible operon. You have to explain why. And then it tells you to draw a diagram for each one. I'm going to make your workload just a little bit lighter. You need to draw one diagram for a repressor and one diagram for an inducer, okay? So I don't care which one you draw it for, but you have to have two diagrams. One repressible diagram and one inducible diagram. So 2 diagrams instead of 6. But go ahead and answer the questions for each. Is it repressible or inducible? And explain why, how you know that, okay? So that'll let me know whether or not you understand these operons, okay? If you get stuck or have any questions on it, then let me know. And if not, then you'll have a great day.